Hi, my name is Sohanjita. I'm the founder and chairman of NoCell, network of researchers on the chemical evolution of life. So today I'm going to talk about one of my papers. So let's go. This PowerPoint presentation is about my paper which was published back in 2021. It was entitled The Way Forward for the Origin of Life, Prions and Prion-like Molecules First Hypothesis. MDPI's journal Life is considering this paper to be the best research paper for the year 2022, hence the video. And before I do anything else, let me talk about the availability of prebiotic amino acid during the chemical evolution of life. My researchers told me that there are a list of 13 amino acids altogether which were available, and one of the amino acid which is really absent will become clear in the story later on is the cysteine amino acid. Next. Let me make some clarification in relation to large molecules. Proteins, peptides, and prions are large-scale structures and are made, processed, and modified in the cells of all cellular life forms. So, such huge structures could not have been present on the early Earth because they would have to have been extremely difficult to make abiotically. However, oligopeptides and oligoprions being small is a better proposition. Finally, we focus primarily on the oligoprions because they are more resilient compared to oligopeptides. Why prions first hypothesis? Either peptides and prions can be small at 7 to 20 amino acids when they take the prefix oligo. The key difference between oligopeptides and oligoprions is that the latter is more resilient due to the presence of extensive hydrogen and ionic bonds. However, oligoprions do not contain disulfide bridges due to the absence of cysteine residues. Cysteine is conspicuously absent in the inventory of early prebiotic amino acids. Please check the previous slide. More will be said about the cysteine and disulfide bridges. Thank you. The ancient nature of nucleic acids, prions, and other related molecules. What we have here are RNA, NAD, FAD, ATP, and acetyl-CoA, which are theorized to be chemical fossils still active within all cellular life forms on the Earth today. Prions and prion-like proteins can also be considered to be chemical fossils. Prions are exclusively made up of amino acids and are therefore proteinaceous with diverse functions in some prokaryotes and eukaryotes. Prions are ancient and are found in all three domains of life, as well as in both DNA and RNA viruses, which contains coding regions for prions. RNA viruses were believed to be present during the very early history of the last universal common ancestor. Prions, like proteins, behave like prions but are not prions. Replication and propagation of neurodegenerative disease are exemplified by beta-amyloid tau alpha-synuclein TBT43 proteins. Prions and prion-like molecules are capable of non-Mendelian behavior by bringing about a type of mutation conferred via transfer of a shape to other normal healthy prions. Prion-like proteins afford protection for newly emerging RNA polymers during the early Earth's harsh environment some 4.1 billion years ago. The interaction between prions with RNAs led to the permanent emergence of relationship in the form of ribonucleoproteins, which are now observed to be nanomachines as exemplified by ribosomes. Thank you. Nature of prions and prion-like molecules. Although I've already made this statement that prions are peptides and can be small, around 7 to 20 amino acids long. Now I'm saying that domain of yeast prion SUP35, which is made from only 4 amino acids, namely glycine, arginine, glutamine and tyrosine, with a length of 7 amino acids. So this is the 7 amino acids strung together, and this particular oligopeptide can self-replicate. Murray reported an oligopeptide made from 5 amino acids. So that would be glutamic acid, serine, valine, alanine and aspartic acid, and in his case there are 9 amino acids strung together, and this oligopeptide can self-replicate too. Prions can be resilient due to the presence of hydrogen and ionic bonds. Cysteine amino acid can form disulfide bridges, but was absent from the prebiotic inventory of amino acids. Prions can help nucleic acids. Example, the ure3 prion helps the uptake of uridosuccinate, leading to the manufacture of uracil. Prions can interact with an RNA effectively to form ribonucleoproteins. And this overlay shows the cysteine amino acid, and it shows here how the disulfide bridges are formed between two cysteine amino acids, and if they're present in a protein, they will help to bind tighter in terms of three-dimensional structures. Thank you. Examples of prions RNA interaction from contemporary biology. Early during this PowerPoint presentation, I've declared that prions are also chemical fossils. I took some examples from contemporary biology of prions to illustrate my points that prions are highly versatile, as exemplified as follows. The adaptive nature of prions, as exemplified by domains of, say, for example, SUP35, may substitute for another, and also prions are capable of serving as building blocks for diverse morphological forms. Many prions exhibit simple behavior phenomenon, hormesis, involved in reversible binary switch, homeostasis and ion regulation. Prions are also involved in RNA amplification and protection. A 35 fragment of the prion-like protein TDP35 binds to RNA and triggers formation of cytoplasmic inclusion that alters RNA processing. You can actually check further information at my paper by clicking here, and this overlay shows that if the poly A is to attach on this prion here between amino acid number 21 and 31, what would happen? A pincer-like loop will be formed, and this will eventually get excised from the main prion, and what would actually happen is that this will get dissolved, leaving behind toxic prion, now activated toxic prion. And if poly A were to attach here, between 21 and 31 amino acid, then nothing actually really happens. It remains non-toxic. Thanks. This is my final slide but one. So if you want to find out more information, please visit the MDPI's journal, and or you can click here to get quick access to it. These are the reference that you, if you want to quote, and you can take a photograph of this. And otherwise, enjoy the read, and I'm sure you'll have fun. Thanks a lot, and bye. Thank you for listening.